Okay. So let me know if you're able to see my screen.
Yes.
Okay,
Yeah.
fine. Today let's do a macro program, which will, so we will do this and we will end listing. Tomorrow onwards, we will start with tables. This is an interesting program. So if you see, there are three tables for adverse events. One is serious adverse events leading to death. Okay. The next listing is just for serious adverse events. And the third listing is for just adverse events. Okay, not serious. Now, if you carefully see, for all these three listings, the structure is more or less the same. We have the subject number, we have a concatenation of adverse event, primary system organ classification, and preferred term. Okay, then what? We have start date time, we have end date time, we have what is the serious event, what is the seriousness of the event, what was the action taken, what was was the adverse event, did it had any relationship to the drug, and what was the outcome as a result of the adverse event. Okay, was it recovered, resolved, what happened? Now, before I go, just wanted to make you understand one thing, that this is the ADAE data set. Remember you were discussing about why we were using ADSL all the time. So now, when the listing is related to adverse event, we have to shift our gears a little bit towards adverse event ADAE dataset. So if you see, this is the study ID, this is the U subject ID, this is the subject ID, this is the site ID. These are the age of the people, this is the ethnicity, country, safety flag, this is the treatment which was given. If it was, this was the treatment, it was fine, this was the drug, this was the placebo, so on and so forth. If I go to the right, this is the all the adverse events which happened to these patients as a result of the study. I mean, not as a result of the study, I would correct my wordings. These are the adverse events which happened to all the people during the study tenure. Some had bradycardia, some had syncope, some had diarrhea, some had vomiting. And all these adverse event terms has to be mapped to some standard terms. Okay, now what do I mean by standard term? Suppose you come to the site and you have an adverse event and you tell the site person that I had a lot of headache with I had a lot of headache which was very painful and it tired me a lot. So you will say this, it will come from your mouth, and this entire thing will be copied in the, this entire thing will be entered in the ETC system. But this is not a standard term. This is a colloquial term coming out. When we go to the doctor, what does the doctor do? We explain them the symptoms, we tell them what happens. He then, if you see, writes a gist about it, the standard words or the symptoms that we are facing. It's basically the adverse event which is happening. That adverse event should be coded to a medical dictionary called MEDRA. This MEDRA dictionary is the one which which will be used as a part of the clinical trial to code the adverse events. So if you see, hypotension is the adverse event which this particular subject has mentioned as a part of the trial. After that, it was coded to the dictionary. See, hypotension. It was then further, hypotension is what part of the body? It is vascular disorder. And if you see, it's basically a layer of one after the other. So suppose the person has hypotension. The lowest level term is hypotension, AELLP. So hypotension is something of a nervous system or whatever vascular system. So then hypotension is linked to which one? The next level is vascular. It is the high level term. It is what called what? Vascular hypotensive disorder. Then it is linked to what? A more broader term, decreased and non-specific blood pressure disorder and shock. Then it is linked to one of our system organs. So we have got 26 to 32 system organs in our whole body, and finally it gets linked to vascular disorders. So we have vascular problems, gastrointestinal infections, respiratory, endocrine. So these are our body systems. So all these adverse event term, whatever they are mentioning, will get ultimately linked to one of the body systems. And that is something which a medical coder does generally. Understood? Got it?
Yeah.
So this session is going to be, I mean, we are not going to be doing this coding, only a medical coder does.
Medical coding there. So this is another role which the medical coder does. Yes. He understands this. Basically, they are from B Pharma. They know about the medicines. They know about these drugs. I mean, they know about adverse events. And slowly, slowly, based on experience and knowledge, they start linking them.
Which variable is the Medra code?
What?
Out of all these, which is the Medra code?
This is the these are Medra terms. If you see AED code, AE
AED code is the okay.
preferred
Yeah.
term. Yeah. So first is AELLT. So we have what? Like we have paracetamol. So
Mhm.
the preferred term is basically, so the lower level term is, I would say, I mean, no, one second. So we have all kinds of paracetamol drugs like Calpol, Procil, Nice, Dolo. So all these are called preferred terms. So we prefer calling them as Calpol, Nice, Dolo, all these things. But
Yeah.
ultimately they come down to what? They come down to paracetamol.
Yeah. So in this example, paracetamol is the Medra code, right?
Not Medra code.
Uh-huh.
Medra terms. These are dictionary terms. Which are dictionary terms? It is AED code. A
Okay.
term is the one which has been spoken or spelt by the patient, or they have
Patient.
said that
Okay.
this is the one. This
Mhm.
has to be coded. Now, what are the? It has to be coded in different levels. So, hypotension is the lowest level, which the person has come and said. Now, what is the standard term for hypotension? Let's take another example. One second. Let's take correct. This is something which has been spoken by the patient. Acute hypertension of unknown etiology. This is a colloquial term. This gets linked to what? Hypertension. This is a dictionary term. If you see AED code, AE bodsis, AELLT, AEPT, AELLT, AEHLGT, AEHLT, all these are dictionary terms. Okay?
Okay. These all come from the Medra, right?
Correct, correct. And
Okay.
this one, AE bodsis, which you are seeing here,
Mhm.
AE bodsis and AE SOC, they are the same. They are the same. Like this is called adverse event system organ classification, and this is called AE bodsis is called adverse event body system or organ class. Same thing. It's called. Okay. So this is the one. So this contains for all the patients. When is the start date? When is the end date? What? And if I go to the right, you will say what is the severity? What is the relation? See, we had the relationship with drug. We have this. Then if I go to the right, what is the outcome? If it's recovered, resolved, not recovered, not resolved. So suppose there's an adverse event which did not resolve or recover. So we have that. What is the action taken? So we have all these details here. Okay.
Okay. So one question here. There is no difference between AESOC and AE bots. Is
But suppose, yeah, you can keep any one of them. Here they have
okay.
kept two, but you can keep any one of them.
Okay.
That's the same thing. Yes, correct. Yeah. So now if you see, so we have, so if you see a serious adverse events, if you see something is serious or not, we would have serious adverse event. Yeah, see, serious. Which of them are serious? See, we have one Y, we have two Y, we have three Y. So we have some serious adverse events. Okay. Now let's check. So first we are giving this lib name library name. We are getting the atom. Now we are creating a macro A. Within that we are establishing five macro variables v1, v2, v3, and v4. Okay. Now we have given data ADE set adam.ade. Same thing. Now if ampersand v1, okay, we are doing what? SPA, CATEX, A term, A bot, AEB code. Why are we doing this? We are doing a concatenation because we have that. See, first we have the U subject ID, which is there. If ampersand V1, what is V1? At the end, we would get that. What is V1? V1 equals to AE SCR equals to Y and AE outcome equal to fit. Okay. So first remember this thing. There are three reports with which we would be generating. Three reports we would be generating from this one macro because three reports are more or less the same. So let's take the first report into consideration. If at serious adverse event leading to death. Okay. So if you see this is the subject number, this is adverse event, primary, class, and preferred term. Okay. So if what we are doing a concatenation of A term, A bot sys, and AEB code, exactly which is here. A term, A bot sys, primary system organ, and AEB code. Preferred term is basically called AEB code. Okay, you will not get AEPT, but you will get AEB code. So we are concatenating it. Next one, what we had start date and time. So keep what we are doing, keep U subject ID, keeping this concatenation, and then we are keeping these things in place, which we already have in a dataset. AESTDTC, AENDTC, AESCR. Just check it, check here. AESTDTC, AENDTC is given here. ADAE, AENDTC. Serious adverse event is AESCR. Action taken is AACN. Relationship to the drug is AREL. And outcome is AEL. So exactly these columns we have taken. So, okay. So here, what we would be getting is run. So ABAE will have some data set. We'll have some records. We will count those records. Proc SQL no print, select count star into NVR from this ADA. Wait. Okay. So we will have some data set in ADA. And this is what I hope you understand this. What we are doing is proc SQL no print, select count star into this is a macro variable. We are keeping this count of this data set. Suppose ADA has 5 records, 10 records, 15. That count we are keeping in this NVR. Okay. And then we are printing. Why are we doing this? Because we anticipate in these three reports, there might be a situation where we will not get any data for one of the reports. Suppose there is no reports for one of, so there no data for one of the reports. Suppose a person has not got any serious adverse event, or a person has not got any serious adverse events leading to death. That is the thing. Okay. So in that case, what will be the count? It will be 0 for that report. If it is greater than 0, if the count of the report of ADAE is greater than 0, then what we do this entire thing, ADAE set, ADAE proc template, then we do this entire thing one after the other. What we are doing is we are doing nothing but the same thing. We are keeping what? Page 1, then now the same trick. Page 1, U subject ID, SPA. SPA is basically the concatenation of adverse event primary. See, exactly we have written it like that. Adverse event primary, preferred term. Then what we have? AESTDDC, AENDDC, AESER, AACN, AEREL, all these we have written. But if NBR equals to 0, suppose any one of the reports does not have any pertaining data in the data set or in the study, then what we would do, there would be a data ADAE text, no participant meets the reporting criteria. This will be the text because we don't have anything to give in the report, right? The report will be, because since we don't have data, all these columns will not be populated. Then we have to give something like this. No participant meets the reporting criteria. And after that, we will have the entire thing same. Title, title 1, a footnote, everything we will be having same. But at here, we would be giving defined text order, that's it. Instead of all those columns, because they won't be there. Now comes the main important part. So we end the macro. Now we call this macro 3 times with varying conditions. So this is for 16.2.2.2 report. What is this? This report. So what is the heading? serious adverse events leading to death. So that's why, see the condition, AESER equal to Y and AE out equal to fatal. Do we have something like that? Serious event Y and outcome is fatal. Let's see. We have serious Y, yes. But do we have outcome is fatal, AE out fatal? Do we have anything? No. Everything is recovered result. So this is exactly the report where we will not get anything. Okay, V1, V2, V3, V4. If you go up for this report, V1, if we give that condition, we have all these reports, we have for this count as 0, then straight away what will the macro do? Straight away skip all these things and go to this part, because NBR will be 0 and it will display this report. Okay, and it will display this report. And what you see ampersand V2, if you see what is V2, V2 is this 16.2 serious adverse event leading to death. This is the title. So title also we have macrotized or provided it in a macro variable. So we don't have to write it. So that's the beauty of macro, right? We don't have to write the program again and again and again. Otherwise, these three reports, if you see, they are similar in nature. So if we would not have used macro, we would have written the entire program manually. One report for this, another report for this, another report for this. So to avoid that, so this is the interesting part where we are using macro to do it. Now what we have to do, V3 equals to 16.2.2.2. Let's see what is V3. 16.2.2.2. V3 is the hands-on program. This. So we have 16.2.2.2. This is the footnote. Let's see. Have we kept where have we kept V3? We have definitely kept it in the footnote. Yes. No, this is title 3. The hands-on program v3.sas. Okay. Now let's execute and check. So this is for the second report where we have serious adverse events. That's why we have given AESER equal to Y. That's it. And the third one, we have only adverse events where A term not equal to null. So we need to have an A term. So the most decisive factor, if you see, for all these macro variables is this condition. The same is the report, but what is different? The title of the condition is different. One report is talking about serious adverse event leading to death. Second report is talking about just serious adverse event. Since it's just serious adverse events, what we have given, see, AESER equal to Y. And the first report is serious adverse events, but leading to death. That's why AESER equal to Y, seriousness is Y, and also outcome should be fatal. But we don't have anything like that because we don't have any patient that has led to death. And the last is we just want the adverse events. That's why A term not equal to Z, not equal to null. We want all the adverse events of the patient. Now let's execute and check. Suppose we execute the macro. Let me know if you have any questions. Because when I execute, you'll see all the three reports getting beautifully generated one after the other. Okay. I'll execute this. This is the entire macro. So macro, I think, is compiled. Okay. So first you have to compile the macro. Now let's run this and check. See, for serious adverse events leading to death, we don't have. No participant meets the reporting criteria. We have the COVID, we have the protocol, we have the date, we have everything, but we don't have any data for this.
They will not get the headings also, like
Yeah,
those.
correct. We have the headings.
Okay.
The serious adverse event. This is the protocol. This is the date. But within this, no, we will not get the headings. Oh, okay, yes, correct. Correct. We will not get the headings because if we have the headings, then within that we don't have anything, right? That's why this is a standard thing that you need to be doing when you are working on when if any reports does not contain any data as per the study. No participant. If you see, you'll get the footnote as well. But this is something that you need to do. This is something which is a very, very standard report for TLF. I mean, for listings, if you don't have any data for this. Got it? Now let's check the next one.
So where did we include this text that
I can't hear you at all,
the
Shavin.
text that appears?
I can't hear you at all.
No participants. Okay, let me write.
Tell me what's your question? Just tell me a little bit clearly, I'll answer.
The message that says no participants are included in this
Mhm.
listing. Where is that included in the program?
This here. If NBR ampersand NBR equals to 0. So remember where what we did here, the moment we are doing macro ampersand macro. So now if you see this Adam data set, let's see this data set. Okay, let's go to work because this is created. Let's see the ADA data set. This is the work ADA data set. See, no participant meets the reporting criteria. Because since ampersand NBR is 0, ampersand NBR is 0. If you see the log, let's see the log. We have executed, there were 26 observations from the dataset Adam AE. The dataset work.ae contains 0 observation and 8 variables. So it contains 0 observation and 8 variables because there was no data at all for fit, I mean, which had A seriousness equals to Y and the outcome was written. So the content, so the dataset contained 0 records. The moment it contains 0 records, we come down here. If ampersand NBR equal to 0, then do data ADA equal to text no participant meets the reporting criteria, run. Title is this one. This is the title to everything. And here we are giving column text because we already have given here data ADA. So the ADA which was generated, it had all the 0 columns. It had all the 8 columns. If you see the log file, there were 8 variables but 0 observations. So we did the standard processes. You don't have to reflect the columns in the reflect the variables in that report. Instead, what you do, you write something like this: data ADAE, the ones which was created, and in there you give text equal to no participant means the reporting criteria. And that is only you make it, beautify it, make it as per the process here, where you give the title and everything. Then you go down here, proc report, column text. The moment you write column text, define text order, you are doing it absolutely correct. I mean, because proc report data equals to ADA, on that you are doing it. And then you give the style, the header, and everything like this. And we are done. That's why we get the report. Got it? This part wouldn't have been necessary only if there were some data which was there. But it would, but in a standard program, you have to write this part as well if something is zero, if an NBR is zero. Because there might be a lot of listings and lot of tables which would be there. In that case, you don't know, right? Where will you get some data or not? You will not get some data. So this part is an important part. So this is the interesting part because the previous things which we discussed about in listings, it were normal listings which we were doing. But here, what is the effort saving? With one program, if you write it through macros, you just call that program 3 times with different conditions and you get 3 reports generated. That is why macros is needed, and that is the reason why you all had macros. You all learned macros. Okay. Next, let's see this one. Seriousness equals to y. What happened? You did not execute. Variable layout is not in the work.file.ada, or it is ARN is not report definition variable. Page one is not on the file work.ada. Okay, probably I have to execute the macro again. AREL AE out. AREL. Let me execute this once. E1 STR A this one. This is the one. Subject number we have serious adverse events. We had 3 serious adverse events, remember? That has come up. Subject number, adverse event, primaries. Now this is something which you can correlate here. So remember what we did? We actually concatenated the adverse event COVID-19 pneumonia. This is the adverse event. Then we had the primary system organ. What was that? Infections and infestations. Then what we have? Preferred term, which is AED code, again, which is COVID-19 pneumonia. So we have concatenated, and that is exactly which column? This one, SPA. That's why we have given catex. This is concatenation. You concat A term, then there is a slash, then AEBOTSIS, then there is a slash, then AED code. Got it? Then we have rest is the same. AE start date. This is the start date. This is the end date. This is a serious event. Is Y. Action taken is what? Drug withdrawn. So the moment we had the adverse event, the drug was withdrawn. Relationship to the drug, unlikely related, not related. So this adverse event, COVID-19 pneumonia, did it happen because of the drug? What was the relationship? It was unlikely related. It may be, may not be. And this was definitely not related to the drug. That is happened to this COVID-19 pneumonia. And what was the outcome? Everyone got recovered. This is the second, which we have only dealing with serious adverse events. And the last one, which we are dealing with is only adverse events. We are dealing with only adverse events, which we have, where we have adverse events, people having adverse events. Let's check this. Yeah. See, all the data we have for this, because we have to have all the data. This is the first patient where we have hypotension, vascular, and all these things. Start date, start time, serious adverse event, no. Action taken, dose not changed, possibly related, recovered result. So everything you get.
So
What?
Archit, hello.
Tell me.
So there is a no space in between this adverse event and the start date time. So that is fine in the report, like in the
Have the
listing.
space. We have space here.
Okay, in somewhere, yeah, in
That
some
is
part
because
there is no.
so probably we have to adjust this length here. You know where to
Okay.
adjust the length. Go here, make the changes here. In SPA, what we have done 20-20, make it 30-30, and the rest as 10-10-10-10-6-6, something like that. Make this 30. Then if you make this, because this is the best possible way that we can give. So the first one, you subject ID, we have given 15. We can make this 10 and make this 25. And then you check, we can get that done.
So I have one more question. Like there is a, can you go? So what is the like use of this escape care? There is
This one,
the,
ODS escape character.
yeah. Oh, okay.
This is the one. So if you see that, that is the one which we have the line. If you see this line. So what we are doing, the line below, here if you see, compute before page you need a line, after page you need a line. But if you see line at the rate 1, this escape character helps you to understand that this is not only text. This is basically the form in which we want this line. It will be a straight line. So what would be the style? The output width of the line would be 100%. That means it would cover the entire page. The border top width will be 0.25 point. So now that's what I'm saying, that when you practice this, make these changes. Border top width, make this 5. When you make this 5, what we will see that the width of this line has gone very broad, dark. I mean, the width has increased. But escape care helps you to understand that, boss, this is something which is not text. We need this form of a line. I mean, we need this line structure in this way. Like this line would be 100%, the border width would be like this. The moment you remove this ODS cat, this text will come up in this. The text will come up in here. But we don't want a text, right? We want this line which is there. So that is the requirement. Or that is basically the instruction that we are providing to this. Got it? Any questions?
Yeah, got it. So, for this, for the title, you create the macro variable, right? Uh,
Or not
for the
title, not title. The entire report we create macro variables and macro. So
okay,
entire report we are creating.
okay, okay.
And
I
with
see.
one report, we are sufficing these three reports because all are from adverse event. 2.2, 2.3, 2.4. That's why we have written one macro compilation, we have written one macro code. But when we are calling that macro, we are giving different types of variables and calling it so that these three reports are created. Suppose there were two more, when we would have added two more, I mean, we would have called the macro variable two more times and uh, given these input values differently based on the report or based on the based on the report or based on what exactly the report is wanting, what is the condition they are wanting. So first report, so based on, understanding the report is what important. Serious adverse events leading to death. So it is AESER equals to Y and AEQ equals to fatal. Next is what? Next is just serious adverse events. That's why AESER equals to Y. Next is what? We have only adverse events we need. Then what is the thing? We have adverse events. This is the one. Adverse event not equal to null. These are the three main reports you will come across whenever you go to a real study. Also, this is the three main reports you will come across. So you'll get an idea immediately. You can use this program, work it out. Okay. Now when I go back to another report, I won't explain you. Do it at home. Look at this one. This is for lab. Same thing. Subject number, test, visit, high range, user time, result flag. This is for abnormal hematology. And what do we have for the next one? 16.2. One second. This is vital signs. Okay, that's not a problem. Vital signs 16.2.1.7. 1.7 into 1.7. 16 to 1.7, do it. You can do it at home. This one, 16.2.1.6. But if you see here, 16.2.1.6 abnormal hematology values. 1.5 is there. One second. See, abnormal biochemistry values. This is for biochemistry lab, where you have subject number, test, visit, normal range, date, result flag. And again, you have the same 16.6 abnormal hematology values. Now let's go to ADA and see how is it coming up. We already have that. Adam, we go to ADLB because this is lab. Do we have something like this? Biochemistry and hematology? Yeah. See, hematology is packet 1, chemistry is packet 2. So for one, we have given what? If you go down, same thing. One we have given for chemistry, one we have given for hematology, one we have given for methaglobin, one we have given for cytokines. So here, how many actually in reports, how many kind of reports you get? One is for hematology and one is for biochemistry. But in this actual data set, what you see? Hematology is there, chemistry is there, methaglobin is there, cytokines is there. So you work on it. I'll share with you the code. I'll share with you this data set. Sit, understand the report and work on it. It's saying abnormal. If you see the abnormal reports, then what? If PARCAT is this and ENRIND not equals to V6, what is V6? What is V6? Not normal. See, we want abnormal values. Okay. So where we do ANRIND? Where we give ANRIND? Let's see. See, normal, 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 high, normal. We don't want normal. So ANRIND not equal to. Not equal to normal for all, because we want, if you see, abnormal hematology values. So what are the abnormal values for hematology, for biochemistry, for cytokines, for methemoglobin? If you can get that, that's the best part. And everything else is there in the report. You'll see What we will take, normal range is high and low. So you have to concatenate ANRLO, low range and high range. You have to take your subject ID. You have to take the param. You have to take the A visit. You have to take the normal range. You have to take the high and low range. Then you have to take the measurement, ADT, date and time of measurement. Then you have to take the result. What is the eval? And then the flag, normal, abnormal. That. I'll share with you this, and you try it at home. And I'll also share the vital signs one. Try it at home. From tomorrow onwards, we'll start with tables. This is what you need to do. What you need to do in the sense, I'll stop recording, okay?